Silda and Arya, both of my students, are going to present together. Okay, Silda, please go ahead.
Hi, everyone. Let's start this again. I'm Selda Omar.
And I'm Arya Shah.
We are seniors at Shrewsbury High School in Massachusetts, and we're here today to present our summer research project called Flow Cytometry-Based Immunophenotypic Analysis of Human and Marine Cells.
We are incredibly grateful for this opportunity and are excited to share our findings with you.
Okay. Our summer research project was structured to answer the central question in immunology: how could we use the powerful technology of flow cytometry to precisely identify and analyze the different types of cells? To address this, our program was designed as a progressive journey moving from foundational theory to practical applications. For the first 2 weeks, we focused on building our theoretical knowledge of flow cytometry and our immunology through in-depth literature review and comprehensive training on cell engine analysis software. We then transitioned into practical phase of applying these skills to real-world samples in weeks 3 and 4. Our work began with the mouse spleen samples, where we were responsible for data quality control and developing a gating strategy to identify dendritic cell populations. We then moved to human blood samples in week 5 and 6, where I established the gating protocols to identify key cells types and specifically T and B cells. The core analysis for the human samples were performed in weeks 7 and 8, where I carefully examined the expression of pStat3 and pStat5 on markers on T cells across various treatment conditions. I dedicated the last 2 weeks on finalizing and preparing for this final presentation. Let's begin with an overview of the immune system. Immunology is the study of how our body protects itself against diseases. It focuses on immune system, which is the network of specialized cells, tissues, molecules that work together to defend us from harmful microbes like bacteria, viruses, and fungi. There are 2 main branches of immunity. The first, innate immunity, which is our body's first built-in defense. It acts immediately, but it is not specific to a particular germ. This includes barriers like the skin, as well as cells such as macrophages. The second, adaptive immunity, it takes more time to accentuate, but it is very precise. Adaptive immunity targets specific invaders, and it also creates immune memory. So if it were to encounter the same pathogen again, the body can respond much faster and more effectively. Together, these 2 systems give us both immediate protection and long-term defense. Okay. Flow cytometry is a technology that allows us to quickly and accurately analyze many properties of single cells while they are in suspension. It uses laser technology to give us information about each individual cell as it flows through the instrument. The principle behind this method is fluorescence. When a laser excites a fluorescent molecule of fluorochrome, the electron absorbs that energy and then releases it back as the light as they return to their resting state. To target specific cells, fluorochromes are attached to antibodies, where then they bind to antigens on the surface of the cells. This means we can label and track different cell types because of the different fluorochromes emit different colors. Therefore, we can study several markers of the same cells at once. Okay. Flow cytometry has 3 main components: fluidics, optics, and electronics. The fluidic system handles the flow part of the flow cytometry. Its most important job is to ensure that every cell is analyzed individually, and it does this through the hydrodynamic focusing, using the fast-flowing sheath fluid to compress the sample stream to force the cells into a single file line. This is crucial for getting accurate measurements and prevents the cells from clumping or passing through the laser at the same time. The next is the optic system, is where the cells are actually analyzed, and as each cell passes through the laser, it scatters laser light. There's 2 key measurements are taken: forward scatter, which tells us about the cell's size, and side scatter, which gives us information about the internal complexity. Most importantly, fluorescent dyes are attached to the antibodies of the cell, which emits light when excited by the laser, allowing us to identify specific cell types. Finally, electronic system translates the light signals to digital data, which uses highly sensitive detectors like photomultiplier tube, also known as PMTs, to convert the weak light signals into electrical pulses. This electronic data is then sent to a computer where it's processed and displayed at a scatter plot or a histogram for analysis. So now that we understand the basics of flow cytometry, let's look at how we analyze the data. I'll walk you through the brief introduction to a software platform called Cell Engine. After selecting files under FCSC, the next step is gating. Gating is a process of drawing a boundary around a specific populations. You can create different types of gates, such as rectangular, polygonal, or freeform to precisely select your cells of interest. Once you gate all the cells of populations of interest you want to analyze, you can create a gate hierarchy. This is a structured series of gates where you can create sub-gates within larger gates. This allows you to progressively narrow down your focus, moving from a broad population to a very specific cell sub. With your gates and hierarchy established, then you can proceed to illustration section. These illustrations are essential for interpreting your findings and presenting your results. Okay, so we're getting to the good part. Our first project was a foundational one. We started with a simple experiment using flow cytometry color to get comfortable with the entire workflow. And this was essential. This was an essential step because it was building our skills before moving into more complex experiments. This project's objective was to identify and quantify dendritic cells in mouse spleen samples, comparing three distinct types: an unstained sample, unpurified, and a purified sample, which was enriched for the dendritic cells. Our first step was data quality control. This was a crucial part of the process. We carefully checked for any artifacts or inconsistency in the raw data, ensuring its reliability, as you can see in the figure below. They show changes in the cell. Next, I performed initial gating. This is the process of drawing boundaries on the data plots to isolate the cells we want to analyze. You can see this. You can see the plot how we identified the mean dendritic cells population based on their unique size and internal complexity. Following this, I performed singlet exclusion. This ensures we are only analyzing the individual cells and not the cellular clusters, which can lead to errors. Then we moved on to the mean analysis. Within our isolated population, dendritic cells, I assessed the levels of CD11 FITC expression. The CD11 marker is a key identifier for the dendritic cells. By looking at the fluorescence of the FITC marker, we were able to identify and tell the difference between CD11 positive and CD11 negative. Finally, I quantified the result. This bar chart provides a clear summary and showing a significant increase in the percentage of CD11 positive and in the purified sample, which validates our purification method.
Okay, I will now explain our second project, which is a multicolor flow cytometry experiment focusing on T cells analysis in human blood. This was a significant step up from our previous work, allowing us to apply our skills to a more complex and clinically relevant problem. On the left, you can see our hierarchy gating strategy. We started with a total cell population and used specific markers to progressively narrow our focus. We first identified T cells using the CD3 marker and B cells using CD20. Since our project was focused on T cells, we created a gate around the CD3 positive population, which you can see here. We used it to characterize CD4. After identifying CD11, we further subgated PSTAT3 and PSTAT5. Our goal was to investigate how different treatments affect the dynamics of these T cells. We exposed the cells to a variety of conditions, including an unstimulated control, two known cells activators, LPS and PMA, and two different cytokines, IL-10 and IL-2 plus GM-CSF. We then measured the expression of two key signaling markers, PSTAT3, which is associated with inhibitory signals, and PSTAT5, which is linked to activation. The histogram on the right shows our key findings. We can see how the percentages of PSTAT3 positive and PSTAT5 positive T cells changed under each condition. As expected, treatments like LPS and PMA caused a clear increase in the activated PSTAT5 plus population. In contrast, the IL-10 treatment led to a significant increase in the inhibitory PSTAT3 plus population, demonstrating its role in immune suppression. Over the course of this project, we gain a comprehensive understanding of flow cytometry principles and data analysis, successfully applying these skills to characterize specific cell types and signaling proteins. Looking ahead, we plan to apply these foundational skills to our future coursework and research, as this project has inspired us to continue exploring the fields of immunology and cellular biology. I'd like to end our presentation by acknowledging and thanking everyone who made this incredible opportunity possible. First and foremost, a huge thank you to Dr. Ruchi Jain and STEM Peers Horizons Quest programs. Their unwavering support and tireless efforts are the reason we are all here today, and I'm grateful for this platform. I'm especially grateful to Dr. Sharma for her incredible mentorship. She patiently taught us the fundamental principles of flow cytometry and its many applications, turning what could have been a challenging learning experience into a truly enriching one. Finally, a special thank you to Anura Ray from Cell Engine for assisting us with our data.
Okay, well, thank you so much for sticking with us and listening to this presentation. It has been a wonderful experience. And to wrap this all up, I'll be happy to answer any questions you may have.
Thank you.
Thank you, Silva. Thank you, Arya. I think we can take 1 or 2 questions. We still have like 2-3 minutes left, so we can take 1 or 2 questions. No questions?
No
means
questions.
means either that everything was very interesting and everybody understood everything, or means that nobody understood anything. So okay, we'll be positive and we'll assume that everyone understood everything. All right. Thank you, Silva. Thank you, Arya.